All right, today we are going to take the proteins that we made last time and assemble them into virus particles. We're going to talk about some general principles of virus assembly using a couple of selected examples as usual. Now, all viruses, all virions, have to undergo a set of reactions to be formed. And these are outlined here. Many of them are in common. The yellow ones on this slide are in common to all viruses. For example, you have to make all the individual structural units from viral proteins. The viral proteins assemble into structural units. You make a shell in some cases, but some cases during budding you're not really making a shell till later, so this is not always the right order. You have to get nucleic acids into the particle, right? This is an important part of virus assembly. If you can't put nucleic acids in there, the infection will not spread to the next cell, obviously. In some cases, the virus particles get an envelope. That's the blue one here. It's blue because not all viruses are enveloped, of course. And then you have to be released from the host cell. Now, sometimes release occurs before maturation or is concomitant with. Sometimes maturation occurs first and then the virions are released from the host cell. So these steps can vary in their order, but they're all steps that viruses have to undergo. And as you know from what we've talked about so far in this course, there are quite a few different kinds of virion particles, although we did arrange them into very specific groups like icosahedral particles with or without an envelope, for example, or with envelopes. Viruses with helical genomes, helical symmetry, and of course among the animal viruses all of those have envelopes. And if you just look at the structure of these viruses, you should be able to know how it's assembled, as we'll see today, how it gets into a new cell, an envelope virus with a membrane that has to fuse somewhere in the cell versus an icosahedral shell, and also how it begins to replicate. Now here's an example of what structure can't really tell us, and that's a comparison of two picornaviruses, poliovirus and rhinovirus. And they have very similar particles. The two particles are shown here. I don't know, can you see any difference between those two? Yeah, there's some subtle differences, but the point is that they're quite similar. In fact, the amino acid sequences are very similar. The genome is similar in size. The capsid looks the same, yet they have really different biological properties. Poliovirus enters the gut. We ingest these viruses. They pass through our stomach on their way to the intestine, which is where they will replicate. So they're resistant to extremes of pH, the low pH of the stomach, the higher pH beyond that, and all the digestive enzymes that it encounters. On the other hand, rhinoviruses, when they do enter the stomach, they get taken apart by the low pH. So they're completely susceptible to low pH. Rhinoviruses are respiratory pathogens where the pH isn't low, of course, but occasionally you do swallow them. That's part of our defense system, is that there's an elevator, a mucociliary elevator in our respiratory tract that brings up virus and mucus, and then you swallow it where it gets digested. Doesn't sound very nice, but in fact that's a good defense system. Or you could spit it out, I suppose, but most of the time we do swallow it. And when this virus finds itself in the gut, it's inactivated. So here we are, two viruses that look, for all purposes, exactly the same, yet there are a few amino acid differences in the capsid that confer this biological difference. So I don't want you to think that just because it's an icosahedral virus that it has certain properties. Now, one thing that you have to remember is that all of the particles we're going to talk about have to be metastable. That is, they have to survive in the wild, whether the wild be, the gut tract or the respiratory tract, but they do have to come apart during infection. This is a concept we talked about during the entry lecture. Metastability, stable under some conditions and unstable under others. Now, assembly is interesting. Assembly happens in a cell, and as you'll see in a few moments, there's a pathway of assembly for all viruses. So things happen in a certain order. And this is typically not reversible in the cell where the virions are being made. But when these new virions reach a new infecting cell, then their stability goes away. That assembly pattern can be reversed. So this tells you right away that where the virus is assembling in the cell is very different from where it is when it enters a new cell. The conditions are different, such that in the assembly cell, assembly is irreversible for all intents and purposes, but reversible in the infecting cell. So just keep remembering that when we talk about this assembly pathway. Like every other aspect of viral replication, assembly also depends exceedingly on the host. Now, the virions encode structural proteins, and for the most part, all the proteins, or the vast majority of proteins in virions are virus encoded. There are very few exceptions. Yet, the virus requires cell functions to put them together into a nice virion. For example, many of the virion proteins have to fold properly. They need cellular chaperones to help them to fold properly. Otherwise, they don't fold in the right way. Cellular transport systems move proteins and nucleic acids to where they need to be in order to form a new virion. As I said earlier, these things just don't diffuse around in the cell. They're actively transported, and we know that from experiments where we use inhibitors of transport. So those transport systems are essential. Secretory pathway is also important for viruses that have glycoproteins or anything that moves through the ER and the Golgi. That has to go through the secretory pathway. And finally, you already know that many viruses replicate in the nucleus. Today we'll see that many of those also assemble. So you have to be able to get in the nucleus and of course get back out again. And that depends on the import and export machinery. So all of these are very complicated processes, and the virus could never encode one of them for themselves unless they were very complex, in which case they wouldn't be viruses anymore. Nothing happens fast in dilute solutions. This reminds me of a saying in a BSL-4, which is a high containment facility where you have to wear a spacesuit to work on viruses. Nothing happens fast in a BSL-4. You have to be concentrated to assemble quickly in a cell, or with reasonable speed. And so this is actually achieved in a variety of ways, concentrating viral components so assembly occurs in a reasonable amount of time. And this leads to sometimes visible bodies in the cell, which we often call inclusions. So that's one way of speeding up assembly, to make these inclusions. Sometimes they're called factories in the case of vaccinia virus and a few others. And here on the lower right is an example of such an inclusion body. This is a neuron in an individual unfortunate to have been infected with rabies virus. And the cytoplasm of this cell has these very distinct sort of round structures. And these are factories where rabies virions are being assembled. So again, the point is you want to concentrate all the components in one place so reactions go faster. And in many cases, these inclusion bodies form something that you can see in a light microscope, which is how this photograph was taken. And they're given names. So many years ago, people would discover them, and then the pathologist typically who discovered them got to name them. So these are called Negri bodies. And if you see Negri bodies in a neuron, it's quite likely that that individual has rabies. So a lot of these are what we call pathonomic for infection. That is, if you see these in certain cells, they are pretty typical of certain virus infections. Other ways of concentrating proteins include using membranes within the cell. You may remember that poliovirus replicates its RNA on vesicles that it induces in the cell on the surface of these vesicles. You may say, why on the surface of a vesicle? Well, it's probably more concentrated than having all the components floating around in the cytoplasm needing to run into one another. So you do it on a vesicle where everything is happening. So that's what I'm talking about when I say concentrating components for assembly. How do we do this? How do we get to a certain place in the cell so we can be concentrated and assemble more efficiently? Well, one way is to do it in a compartment, a factory or some localized site. So those Negri bodies were localized sites, and we don't really understand how viruses form these for the most part. There are some clues, but We don't know what makes a particle go to one place. Probably a protein or several proteins nucleates the formation of a factory, and then it simply grows. Sometimes you have localized production of proteins, and as I said, a single protein is made, then you have protein-protein interactions, and it attracts more proteins, and that makes a factory. And you can also make subassemblies, as we'll see, of individual proteins, which then join to form the virion. Also, the membrane plays a role, as we've seen for RNA synthesis. Structural components often line up right under a membrane. We'll see that a bit later. And you get interactions, protein-protein interactions, that help to form an individual virion. And this leads to so-called membrane patches, where you can actually see before viruses leave a cell. So we'll see later, many viruses leave the cell by this process called budding. And just before a bud forms, you can see right beneath the membrane a patch where there are viral proteins that are being accumulated. So they're being directed to this very one specific part of the plasma membrane.